Okay, I think we should get started. Good afternoon. My name is Yi-Hong, and it's a great pleasure to introduce today's seminar speaker, Meng Wang, from Genelli Research Campus. So Meng got her bachelor degrees from Peking University, majored in biochemistry and molecular biology, and then she studied under the supervision of Dr. Dirk Bowmans at University of Rochester, where she discovered a role for the junct pathways in oxidative stress, and therefore regulating lifespan in C. elegans. And in 2005, she moved to Boston and did her postdoc trainings with Gary Ruvkun at Mass General Hospital, where she continued to use C. elegans as a model organisms to study the lifespan regulation. And she started her own labs in 2010 at Baylor College of Medicine in Houston, and then quickly established herself as a major player in this field. And she got promoted to associate professor 6 years later. And then she was also very quickly promoted to full professor and became the endowed, the Robert 5 endowed chair in the Aging Institute. In 2018, she got appointed as the Howard Hughes Medical Institute investigator. And then just last year, she moved her labs to Genelli campus just across the river. So if you look at her CV, it's very impressive, although she's very young and she has received many prestigious awards. She's clearly a rising star on the upward trajectory. But I think what impressed me most is when I look at her records, and she turns out to be a marathon runner, and she actually could finish marathons within 3 hours and 30 minutes. It's something that I really admire. And so without further ado, welcome, Meng. I'm looking forward to your talk.
Thanks, Yi-Hong, for the invitation and also very kind introduction. Yeah, it's great. You just recently moved into the area, so I really appreciate this opportunity, to visit NIDDK campus, to get interact, with colleague and in this area. So really looking forward like future collaborations and more interactions. Yeah. So as mentioned, since I was a graduate student, I really become interested in this question of aging biology. I think many of us was enter this field motivated, at least for me, was motivated by this question that Aristotle already asked more than 2,000 years ago. Why some animals being long-lived and others short-lived? And given the complexity of the living organism, so you can imagine actually this question can be tackled at many, many different levels and from diverse angles. So actually in the field that exactly what happened, I have colleagues working on environmental regulation of longevity and looking at epigenetic factors, genome instability, cellular senescence, from all aspects. So it's very exciting research areas now. But in my group, we choose to work at the interface between metabolism and longevity. In particular, we are interested in the signaling role of metabolite in supporting cellular homeostasis and organism fitness. So as we know, metabolites are the product from thousands of chemical reactions in living organisms, and their production is dynamically linked with all kinds of cellular activities, and they're also very, very well conserved across different species. And they were best known as structure building blocks, energy fields, and biochemical cofactors. But in the past decade, through many exciting work in these research areas, now we know metabolites are also crucial signaling molecules. So in our studies, we're interested in those molecules, and through the years, we have identified specific metabolite that can actively mediate the communications between different organelles within a cell, and between different cells and tissues in organisms, and even between microbes and their hosts that share the same ecosystem. And using Cinorhabdiasis elegans as model systems, we're able to decipher those metabolite-mediated regulatory mechanisms and reveal their importance in regulating longevity. So we For those signaling metabolite, we kind of focus on two major sources of them, and the one like from intracellular lysosomes and the one from environmental bacteria. So today I would like to share with you some published and unpublished work along those lines. So first, let's look at the lysosome. So Juan's here, so I kind of feel a little bit embarrassed to introduce this very important cell organelle in all eukaryotic systems. So it's very specialized because it constitutes acidic environment inside the cells. It acts downstream of endocytosis and phagocytosis to digest all kinds of materials taken up from the environment. It also interacts with autophagy and responsible for degradation and the recycling of unwanted cellular component. And for those important digestion and degradation functions, lysosomes carry a variety of hydrolases in the lumen and also on the membrane. And their activities are directly coupled with metabolite production from these organelles. Therefore, they are very highly metabolic organelles in eukaryotic system, in addition to like mitochondria and ER. So several years ago, when I was a postdoc with Gary Rufkin, we linked one of those lysosomal hydrolases with longevity regulation in C. elegans. And this is a lipase called lipo4. So how we find this lipase? So that time we were looking for some factors that act downstream of multiple pro-longevity mechanisms, hopefully to find something that we can manipulate and then to have a beneficial effect. So therefore, like we found this lipase through a genetic screens. So in wild-type conditions, its expression level is very, very low, barely detectable, but it dramatically upregulated under several long-lived conditions that we know at that time, including reduction of insulin IGF-1 signaling, fasting, and the germline deficiency. And then I start my own lab, my first MD PhD student, Andrew Follick, working on this lipase. And then, first thing he look at is to look at subcellular localization and find actually this lipase is localized at the lysosomes and also is carry acidic lipase activities. And he further made a transgenic strains to constitutively express this lipase in the intestine of C. elegans, which is their major fat storage tissue. And you find those transgenic strains show in the red curve live 40 to 60% longer than the control ones show in the black curve. And then he further delineates the lysosome to nuclear retrograde lipid signaling pathways that mediate the longevity effect here. So since this work published several years ago, only show the model here. So basically what we found is when this lipase is induced in the lysosomes, and it will promote the translocation of a fatty acid binding protein called LBP8 from the lysosomes to the nucleus. And the fatty acid binding proteins at the structure shown here, like we solved it with Eric Altland's group a few years ago, it had this lipid binding motif, and then it served as a lipid chaperone. So basically bind fatty acid and their derivatives and facilitate their transport into different cellular compartments. And in our case, we find this lipid chaperone LBP8 combined to a fatty acid derivative called N-oleoyl-acetanolamine, OEA. So we find this OEA is increased when this lipase is induced in the lysosomes, and it's directly bind with LBP8 proteins. Then LBP8 protein can facilitate its nuclear transport. And then in the nucleus, OEA combines to the downstream nuclear hormone receptor, which is NHR80 in C. elegans, and then it works together with another nuclear hormone receptor, NHR49, to regulate the downstream transcriptional responses and then promote longevity. So upon the discovery of this lysosomal signaling, then we start to wonder, whether the OEA can be directly generated at the lysosomes, because no one knows, And then also we wondered, okay, when we increase lysosomal lipolysis by overexpressing lipo4 in the intestine cells, whether or not will affect the protein composition of the lysosomes and to accommodate the signaling effect. So with these two questions in mind, former postdoc Yong Yu, who now started his own lab, adapt the lysosome immunoprecision method in C. elegans. So this allowed him to purify both lysosomal and paired non-lysosomal fractions at the same time from adult C. elegans. And then working with two current graduate students, Max Gao and Yu-Chen Guan, they profiled the proteomes of both lysosomal and non-lysosomal fractions using mass spec. Then through comparative proteomics analysis of these two paired fractions, they were able to define what they call lysosome enriched proteomes. So then after they established this method, they apply it to compare the lysosome enriched proteins between wild type and then the long-lived lipo4 transgenic strains. The first thing they find is compared to wild type lysosome, the lysosome from the lipo4 transgenic strains recruit many more proteins to these organelles. So suggesting by increasing lysosomal lipolysis, the lysosome protein composition will have a dramatic change. So it kind of, that time, I was surprised by this result because I always thought the lysosome protein composition is static. So, you learn this list of enzymes and the membrane protein should be always there. But this kind of show actually is more dynamic than we thought. And so I think make it very interesting to further explore these areas. And then from this list, we did find 3 NAP phospholipase D enriched only at lysosomal fractions. So it's known that NAP phospholipase D is required for the biosynthesis of N-isoanolamine, including OEA that I mentioned to you. So this is a class of fatty acid derivative, and these enzymes are required for its biosynthesis. So then therefore, the enrichment of those enzymes on the lysosome suggests actually this class of fatty acid derivative maybe can directly generate it from the lysosomes. And this was not known before. So now we are doing more biochemical analysis and the genetic studies to really confirm it. At the same time, when we look at this long list of protein candidate, we also got unexpected findings. So as shown here, in wild type, actually we didn't see a nuclear protein enrichment on the lysosomes, but they are significantly enriched in the lysosome from the lipo4 transgenic strains. And then 2 of them are nuclear porine proteins, which is a part of the outer ring of the nuclear pore complex. So based on this result, we hypothesize maybe there's increased interactions between lysosomes and nuclears. So therefore, we see a higher enrichment of nuclear proteins. And to test this hypothesis, we generate a strain and that express lysosome LAMP1 tagged with RFP that allow us to visualize the distribution of those lysosomes in intestinal cells of adult C. elegans. So we find in wild type conditions, lysosomes are more like evenly distributed in the cytoplasm, but in the lipo4 transgenic strains, we see a higher clustering around the nucleus. And then Max further wrote a small script that allow us to quantitatively measure this change. And we find indeed in the lipo4 transgenic strains, there is a shift towards the paranuclear regions. Then the question is whether this positioning change contribute to the longevity regulation. So we have knocked down the NPP6 by RNAi and find it can fully suppress the lifespan extension caused by the lipo4 transgenic strains. But when you look at the lifespan of the NPP6 RNAi, you may wonder whether the suppression is due to non-specific lifespan shortening because they are much shorter lived than one type animals. So we have the same concern. So therefore, we also knock down MPP6 in other long-lived conditions. So I'm only going to show you the result from the DAF2 mutant, which reduce insulin IGF-1 signaling. So you can see this mutant actually have very long-lived, double the lifespan. But when we knock down MPP6 by RNAi, it still shortened the lifespan, but it does not affect the lifespan extension at all. So it suggests that the longevity effect that we see with MPP6 is not just because it makes animals short-lived. And consistently, we also see in the DAF2 mutant conditions, there's no paranuclear accumulation of the lysosomes. So together, it suggests us that the increased interactions between lysosomes and the nucleus might be specific to the lipo4 induced lysosomal signaling. And same time, when we look at the proteins that enriched, specific enriched with the lipo4 transgenic strains, we also find two AMPK catalytic subunits, so AAK1 and AAK2. So as you can see here in wild-type conditions, AAK2 and AAK1 are only detected in the non-lysosomal fractions. But in the lipo4 transgenic strains, the AAK2 are detected in both fractions, but it's more than 10 times enriched in the lipo4, in the lysosomal fractions. And for AAK1, actually, it's only detected in the lysosomal fractions, but not the non-lysosomal fractions. In contrast, if you look at the DAF2 mutant, another long-lived conditions, the AAK2 is detected in both fractions, and AAK1 is not detected. So based on this result, we hypothesize that upon the lipo4-induced lysosomal lipolysis, the AMPK might be recruited to the lysosomes for its activation, and this may contribute to the longevity effect. And in supporting this idea, when we knock down both AAK1 and AAK2 at the same time, it can suppress the lifespan extension in the lipo4 transgenic strains. So together, although we still need to do a lot of work to support the model, but this is what we have now. We think this positioning change of lysosomes did have a functional importance. So the increased proximity between lysosomes and the nuclear pore complex, a nuclear pore may facilitate the lysosome-derived metabolite into the nucleus, and then in turn facilitate their signaling effect on regulating transcriptional responses. At the same time, those paranuclear accumulated lysosomes may also provide a platform for recruiting nutrient sensing factors like AMPK, and then help to trigger the downstream signaling effect. And we think both maybe contribute to the longevity effect in these transgenic strains. So together, those work kind of suggest that lysosome-derived metabolite can mediate the crosstalk between different organelles. So the example I show here is a lysosome and the nucleus. Then next, what we want to know is whether or not lysosome-derived metabolite can also mediate the crosstalk between different tissues. And then so a former graduate student, Mazia Savini, is interested in this question, and she got her first hint from the transcriptal analysis of these long-lived transgenic strains. So she finds, interestingly, the neuron peptide signaling pathway is overrepresented in the lipo4 transgenic strains. So as shown here, in C. elegans, in the nerve system, the neuron peptide is synthesized and then processed from the pre-pure protein and cleaved and emanated into their mature form. And for all the enzymes involved in this process, highlighted in red colors, their encoding genes are upregulated in the lipo4 transgenic strains. So, and then furthermore, through the RNA-seq analysis, we also find over 30 neuron peptide encoding genes are also upregulated in these long-lived conditions. then the question is whether their specific neuron peptide are involved in this regulation. So Mazia knocked down all those neuron peptide one by one, and then identify one specific neuron peptide called NLP11. So when it's knocked down in the neuron-sensitive background, it fully suppresses the lifespan extension caused by this lipase overexpression. But if you knock down NLP11 only in the intestine, it does not affect the longevity effect. And conversely, we can increase the NLP11, the neuron peptide gene expression only in neurons, is sufficient to prolong lifespan. But if we only overexpress it in the peripheral intestine, we do not see any lifespan extension. So together, it shows that when we increase this lysosomal lipase, lipo4, in the peripheral intestine, the metabolic tissues, we can specifically upregulate the neuron peptide pathways in the nervous system. And then their specific neuron peptide actually are upregulated and mediate the longevity effect. But the question is, what could be the endocrine signal that mediate the crosstalk between the peripheral intestine and neurons? So Mazia thought maybe the free fatty acid could be the signal here. So since lipo4 is a lipase, it may upregulate the fatty acid signal here. So to test her idea, she has used the protocol that we developed to purify lysosomes from adult worms. But then now instead of doing proteomics analysis, she used those purified lysosomes for lipidomics analysis. And then what she finds is in those purified lysosomes from the lipo4 transgenic strains, the triglyceride species are decreased, which supporting its lipase activities. But more importantly, we find the levels of polyunsaturated polyfatty acid PUFAs are upregulated. Now the question is, are those PUFAs involved in this endocrine regulation of neuron peptide genes? So to test this, again, back to the genetic approach, so Mazia knocked down the fatty acid desaturase called FAT1 in C. elegans, which is required for PUFA biosynthesis. And then she find when FAT3 is gone, when PUFA biosynthesis is blocked, now the induction of neuron peptide genes are completely gone. And then next, she wants to know then whether there are specific PUFAs involved in this regulation. So what she did is she gave individual PUFAs back to the lipo4 transgenic strains with the FAT3 mutant. And then she find only DGLA, the dihoma-gamma linolenic acid, is sufficient to fully restore the induction of neuron peptide genes in these conditions. And consistently, only DGLA is sufficient to restore the lifespan extension in the lipo4 transgenic strains with the FAT3 inactivation. So together, they show that DGLA, the fatty acid, PUFA, fatty acid is an endocrine signal that link the peripheral intestine and the neurons. But DGLA itself is very lipophilic molecules. So it'd be very difficult for it to migrate on its own from one side to another side. So we are searching for factors that could facilitate a signaling effect. So from this genetic screen, Mazia find another fatty acid binding proteins called LBP3. And then here show you some of the data that make us believe that LBP3 is a carrier for DGLA. So what we did is working with Eric Altland group at Emory University, we first profiled the liposomes from C. elegans, Y-type C. elegans. So you can see here, when we look at the percentage of those free fatty acid, the EPA take the largest portion, so close to 20%. But if you look at the percentage of a DGLA, it takes less than 3%. But if you look at the fatty acid, the PUFAs that bind to the LBP3 protein, now we can see the DGLA take the largest portions. but EPA and AA take a very small portion. And on the other hand, if we look at another fatty acid binding protein, which is a close homologue of LBP3 called LBP2, but it does not involve in the neuropeptide gene expression regulation, and we found it showed very, very little binding with DGLA. So it suggests this LBP3 fatty acid binding proteins does have a clear preference towards DGLA as its lipid target. And furthermore, genetically, we find when we remove LBP3, the chaperone proteins, now DGLA lose its rescuing effect completely. Again, suggesting DGLA is required for, LBP3 is required for DGLA to have the regulatory effect. And conversely, we can also overexpress these chaperone proteins only in the intestine, and we found it's sufficient to induce the neuropeptide gene expression. But if we switch the lipid binding domain between LBP2 and LBP3, now overexpress the chimeric proteins lose the effect to induce neuropeptide gene expression. Consistently, we can increase LBP3 levels only in the peripheral intestine. It's sufficient to prolong lifespan. But if we overexpress the chimeric proteins, it cannot promote longevity. So together, suggesting the binding between LBP3 and the DGLA is very crucial for its regulation of neuropeptide gene expression and in turn longevity. So now we think the peripheral derived LBP3 and DGL signal can regulate neuropeptide signaling pathway in the nervous system and then regulate longevity. But to fulfill this long-range regulation, we'll expect that DGLA, LBP3 can be secreted from the intestine and then act on neurons. But how can we test this? So to this end, Mazia has designed an experimental system where she made a transgenic strains to express GIP-tagged LBP3 protein only in the peripheral intestine, and then the GIP nanobody only in neuron. So in this design, the neuronal expressing nanobody will serve as a trap. So if LBP3 protein is secreted and then taken up by neurons, we should detect GIP signal in neurons. And this is exactly what we see. So in the MK positive neurons that expressing the nanobody, we can detect very obvious GIP signals. So it suggests that the LBP3 DGL lipid signals are derived from the peripheral intestine and then secreted into the body cavity and then taken up by the neurons. But exactly how it's secreted, how it's taken up for the neuron, we still don't know. We are doing more genetic screens to figure out. At the same time, in the neuron, yes?
Is it possible that this fat tissue cells from a channel of the gap junction directly transport this to the neuron rather than secrete and then take up?
Yeah, so that's a great question. So the question is whether, Yihong asked me to repeat the question for the Zoom audience. Yeah, the question is whether or not the intestine and neurons will form some direct connections. Therefore, the signal can just directly go from intestine to the neurons. So based on current understanding of the structure, interaction between intestine and neuron nerve system in the C. elegans, we don't think that's the possible explanation because people didn't see that. But we are doing ultrastructure study, the 3D ultrastructure now at Genelia. So, maybe we'll see something that we didn't see before, but based on the current knowledge, we don't think that support that model, but we cannot exclude those models either. Yeah. Yeah.
The nanobody is in the cytosol as this LBP3, so you have to cross two membranes. for these different?
Yeah, so Juan's question, like we put a nanobody inside the cytosol of the neuron, so then therefore those lipid signal need to cross two membranes. Yeah, so that's actually was surprised because what we first actually we just put the nanobody on the surface of neurons. Then we of course detect the signal, but same time we also see GFP signal inside the cytosol. So that's actually the experiment I show is the version 2. Then we put it into the cytosol then to see whether it can be up. Then it also confirmed the uptake. So now the puzzle is like how it can, actually cross two membrane and goes in. So that's why we're thinking to do more genetic screens, just find all the exocytosis, exosome, and all the possible related factor to see, what could be the possible mechanism.
This reader, the culture,
Mhm.
should be able to take the condition
Yeah, we would love if we have such symptoms in worms. So actually we don't have.
That's about it.
Yeah. But that's an interesting question because I presented work in a lipid imaging, and so there are people actually working on fatty acid binding protein homolog in mammalian systems. So they always see there's a secretion of those fatty acid binding protein, but they never thought to look at like neuron connections. So they were thinking actually maybe they can do some co-culture system and to see whether or not the similar effect also work. Yeah, there. So yeah, I'm waiting for their update. Yeah.
It's a great story. If you can you, is it possible to deliver DGLA directly to the nucleus? If you could do that, can you then remove the requirement for the lysosome proximity to the nucleus?
Yeah, the question is whether we have some mechanism to deliver DGLA directly to the nucleus to bypass the protein part. The short answer is we don't have such mechanism right now, but that's an interesting thinking because actually now a lot of people doing those caged molecules, then you can kind of like spatially and temporally control their release. But I don't think anyone is doing that for lipid molecules. But generally, definitely we have a group working on those. But that's a great idea. Maybe we should think about some chemical engineering approach. Yeah.
Because how important do you think is the inter-organelle communication rather than delivering the specific lipid?
I think actually, yeah, the short answer, we don't know. So yeah, because we also cannot rule out actually it's LBP3 itself. So the DGL just needed for the conformational change and therefore it regulates the transcriptional response. So, and all maybe DGLA like the OEA case can directly bind to the downstream nuclear hormone receptor. I think both possible. And yeah. So yeah, so that's actually back to the point that in the nuclear, what happened. So again, we don't know the full story. So only through genetic screens, we know the nuclear hormone receptor NHR49 acts in neurons to perceive these peripheral lipid signals and then regulate neuron peptide gene expression. But again, it's whether the lipid binding protein mediating this changes or the lipid itself as a ligand to mediate this, we don't know. So yeah. So again, this is the area we are still working on. And same time, how the neuron peptide NLP11 signal back to the peripheral. We're also working on the mechanisms, screening the receptors for this neuron peptide, and hopefully we can fully understand this regulation. But this kind of more recent work highlight the importance of lysosome-derived metabolite in coordinating the crosstalk between different tissues. It also highlights the role of lysosomal signaling in systematic regulation of longevity. And this systematic effect is further supported by our recent cell atlas studies. So although people now doing single cell RNA-seq everywhere, but for adult worm, it has been a challenge in the past because they have these harsh cuticles. So it's making isolating single cells very difficult. So therefore, former postdoc Yu Si also now has her independent lab, work with Max Gao, and then collaborate with Hongjie Li's lab at Baylor and develop a single nuclear RNA-seq pipeline for adult worms. And so with this pipeline, we're able to profile the transcriptome changes at a single cell resolution during physiological aging process at 4 different ages. And that allow us to build the cell atlas for worm aging. And then we already put the data, kind of help people to explore those data on this website. It's a alpha version, it's very slow. If myself sometimes have a hard time to get on because now so many people are searching it. So we got the feedback, so we know this is an issue. So we are now working with Genelia to make this portal faster and also add more features for, that you can really visualize all the data that generated from this atlas. So if, for C. elegans people here, if you have any feedback for this portal, just let me know. So because we are really working on the beta version right now. But importantly, with this data, it allows us to build a transcriptomic aging clock using the lasso regression models for different tissues. So you can see the correlation factors is very good for all tissues. And so with that, we're able to predict the biological age of different tissues at the chronological age 6, day 6, which is like early age for worms. So we can see actually all the tissues age slower in the lipophil transgenic strains, although the lipase is only upregulated in the gut, the intestinal tissue, but all the tissues now age better. So it really suggests by upregulating this lysosomal signaling, we can have systematic beneficial effect in multiple organs. So together, those work related to lysosome really make us believe the lysosome metabolism and signaling are coupled, which is also supported by many exciting research now in the field. So we think by changing the lysosome metabolism, there are specific metabolite can function as a direct signaling molecules to mediate the tissue and organelle crosstalk. At the same time, change lysosome metabolism can dynamically influence the protein composition of the lysosome, and then in turn affect its signaling effect. So we think both mechanisms will contribute to organism fitness and longevity. So this is for our lysosome side. So if more questions, if not, I will switch a gear to talk about bacteria host interactions. So in the second part, I will focus on the bacteria side and tell you first, how we discover this communication mode between bacteria and host mitochondria, and then how these unexpected findings inspire us to search for more those bacteria-derived metabolite involved in this communication. So I think now the idea that microbiota influence host physiology and pathology is not new anymore. So I think I see those exciting work almost every month. So now through phylogenetic analysis, different bacterial strains have been connected with host longevity. But more than 8 years ago, when I'm reading those very exciting literatures in this field, I could not stop wondering, then how about same bacteria when they express different genes at different time? So for example, I like both Coke and Pepsi, right? When I drink them, the same bacteria inside my gut may turn on different gene expression and generate different metabolite. So as a geneticist, I was just keep thinking, how this bacterial genetic heterogeneity impact host longevity. So I was very lucky because my first postdoc, Bing Han, who now also has his own lab, also interested in this question. And he said, I want to address this systematically. So we established a collaboration with Christoph Herman's group at Baylor. So Christoph is a bacterial geneticist, and his lab carried a complete collection of all E. coli knockout mutants. So then we screened this full collection. Then out of nearly 4,000 E. coli deletion mutant, we identified 29 that can prolong the lifespan of C. elegans. And then Bing worked on 2 of those bacterial factors called HNS and lung. It turned out they both negatively regulate the biosynthesis of colonic acid in E. coli. So colonic acid is a polysaccharide secreted from E. coli when they are stressed, and its biosynthesis requires more than 19 enzymes that are located in an operon, and HNS and lung can actively regulate this operon. So here I show you the structure of a colonic acid. I hope you can appreciate how complex this sugar is. So it's amazing that E. coli can do the work and just synthesize it from scratch. But the problem for us is then we cannot buy it, or we cannot synthesize it in the lab. So Bing just made a lot of effort and developed protocol that allow him to purify CA from cultured bacteria, and then confirm that purified CA is sufficient to prolong lifespan in both wild-type worms and then also wild-type fruit flies. Then, how this bacteria-derived CA target on host to promote longevity. We're still working on the mechanisms, but through genetic analysis and cellular studies, one mechanism we find is that CA actually can promote mitochondrial fission in intestinal cells of C. elegans and to promote longevity. So again, this work is published, so I'm only show you the model here. But with this finding, we were very excited because that kind of show there is a chemical dialogue between bacteria and the host mitochondria. And based on symbiogenesis theory, we know they are Asian relatives. So it's quite exciting to think that bacteria can still talk to their eukaryotic cousins using their secreted metabolite. Then the question is whether CA is a unique example for such communication, or we just reveal the tip of the iceberg here. So we asked two questions. So first, whether or not there are other bacteria also talking to host mitochondria using their secreted polysaccharide. And secondly, are there other bacteria-derived metabolite other than CA that can signal to mitochondria? So actually for the second question, current postdoc Meng is doing a high throughput screen. So we just finished the screen last week. So based on the result, now I'm confident to say that CA is not the only example for such communication. There are many more bacteria-derived metabolite can also mediate this dialogue. But the exact mechanisms, I think hopefully in the next few years we can figure out. But for the first questions, I discussed this with Bonnie Bassler at Princeton University and Carl Gross at the UCSF. So Bonnie worked on the Vibrio cholerae strain, is kind of they call C. coli. So it's like ocean. And then Carl worked on Bacillus subtilis, the gram-positive E. coli bacteria. So actually in both species, they have actually extracellular polysaccharides secreted. And then they generously share the strain that can manipulate those extracellular polysaccharide secretion. And Meng examined their effect on C. elegans mitochondria and find very interestingly for both strains that increase extracellular polysaccharide secretion, they promote mitochondrial fusion. So this is an opposite effect to CA. But this result make us hypothesize that bacteria-derived extracellular polysaccharide may be broadly involved in the communication between bacteria and the host mitochondria, but they can drive the mitochondrial fission fusion to different directions based on their origin. And we think this difference is associated with the sugar composition in those polysaccharides. So now we are doing a more biochemical analysis to really confirm this is the case. So far the study I show you, we focus on mitochondria in the intestine, the gut cells, where they directly encounter E. coli or bacterial strains. And then the question is, how about other tissues? So in the next 10 minutes, so I will tell you some our very recent unpublished work focused on mitochondria in the oocyte. And this has been leading by a current graduate student, Yi-Tang Li. So I mentioned to you like C. elegans, good system to study somatic aging, so we can follow their lifespan. But actually, it's also a pretty good system to study reproductive aging. So one way to look at it is to use a reproductive lifespan where we follow the self-fertilizing hermaphrodite over time and then score the number of reproducing worms every day. So you can get this reproductive lifespan data. Another way is to use late life fertility, where we mate the hermaphrodite at different age with young males that provide fresh sperm, and then score the number of reproducing worms. So using both assays, we look at worm reproductive aging under different bacteria strains. And we found actually they show a pretty dramatic difference. So as you can see here, for C. elegans on the B-type E. coli, OP50, they reproduce much longer and better than the worms on K12 E. coli HT115. So as we know, the reproductive ability, to determine that the oocyte quality is a rate limiting factor. And actually in our body, oocyte have the largest number of mitochondria compared to any other tissues. So then these findings make Yi-Tang wondered whether the mitochondria in the oocyte will be affected by different bacterial input during the aging process. So he has used the germline specific TOM20 GFP reporter line to examine the mitochondrial distributions in the oocyte. Specifically, and then find actually at a young age, the mitochondria are evenly distributed in the cytoplasm of oocyte on HT115 E. coli, but those mitochondria become hyperclustered around the nucleus upon reproductive senescence at day 5. On the other hand, if we look at the worms on OP50 E. coli, we didn't see this age-related increase in the paranuclear accumulation of mitochondria in the oocyte. So this is consistent with the result I show you. The worm on this E. coli will reproduce longer and better. So now the question is, okay, if we then prevent this paranuclear accumulation of oocyte mitochondria, can we see a reproductive longevity? So to this end, Yi-Tang generates the transgenic strains that overexpress DRP1, the dynamic-related protein 1, only in the germline. And then he finds in these conditions, now the paranuclear accumulation of oocyte mitochondria in the oocyte is completely prevented. And then importantly, in these transgenic strains, worms reproduce much longer on either HT115 or OP50 E. coli. So this shows that different bacterial input can indeed affect the oocyte mitochondria distribution and in turn the reproductive aging in the host. Now the question is how? So on the bacteria side, we know these two bacteria strains carry many different metabolites at different levels. And one important one is vitamin B12. So in HT115 E. coli, they have plenty vitamin B12, but OP50 E. coli have very, very little. So Yi-Tang have supplement the bioactive vitamin B12 to worms directly and find it will suppress the reproductive lifespan extension caused by OP50 E. coli. But if you look at the effect on the worms on HT115 E. coli, they already have plenty vitamin B12. It does not affect the reproductive lifespan there. And consistently, vitamin B12 supplementation also increase the paranuclear accumulation of oocyte mitochondria. So now this make us think that vitamin B12 is a bacterial input that affect oocyte mitochondrial distribution and the reproductive aging in the host. Then what happened in the host? And through genetic screens, we find this involve the succinyl-CoA asensitase in mitochondria. So this enzyme located in the TCA cycle, and it can catalyze succinyl-CoA to succinate with the production of either GTP or ATP. And the difference is determined by the beta subunit. There is a GTP isoform and there are ATP isoform, but it's not by the alpha subunit that's shared by both beta subunit. So Yitang find when he knocked down the GTP specific beta subunit SUCG1 only in the germline, it will lead to reproductive lifespan extension on HT115 E. coli, but it cannot further increase the reproductive lifespan extension in worms on OP50 E. coli. And consistently, germline specific knockdown of SUCG1 also attenuate the age-associated increase of oocyte mitochondria around the nucleus. However, if we knock down the ATP-specific beta subunit SUCA1, it does not affect the reproductive lifespan on either HT115 or OP50 E. coli. It does not affect the oocyte mitochondria distribution at all. So based on those results, Yitang hypothesized that maybe GTP metabolism is specifically influenced by those bacterial inputs and to regulate reproductive aging. So to test his idea, he developed a protocol to purify mitochondria only from the germline of adult worms, and then work with Jay Wang's group at the University of Wisconsin Madison. We measured the GTP and the ATP levels in those purified germline mitochondria. And what we find is, so when we look at the germline mitochondrial GTP levels in the worms on HT115, it's increased close to tenfold with age. But on the OP50 E. coli, this increase is less than threefold. But if we look at germline mitochondrial ATP levels, it does not affect it by age. It does not affect it by bacterial input. So it supports that the GTP metabolism is specifically affected by different bacterial input during reproductive aging. Now the question is whether the bacterial input of vitamin B12 that we find actually act on the CoA synthetase to regulate reproductive aging. And the answer is yes, because when we knock down SUCG1, the beta subunit of the CoA synthetase, vitamin B12 now cannot suppress the reproductive lifespan extension caused by OP50 E. coli. So this together support the model that during aging, the GTP levels in the germline will be elevated, and this will be further enhanced by vitamin B12 derived from the environmental bacteria. And this will result in paranuclear clustering of oocyte mitochondria and the reproductive declines in the host. But if we can reduce the vitamin B12 availability from bacteria in the environment, then we can attenuate these age-associated elevations, and this will help to lead to reproductive longevity. So this data and also, some of the work that I just mentioned, we just finished, started recently, really suggests there is a very interesting communication mode between bacteria and host mitochondria, and they are very important to regulate both somatic and reproductive longevity in the host. So together, I think that those findings early and current recent findings really make us believe that metabolites carry crucial signaling roles across the scale of organisms. So inside the cells, it will mediate organelle crosstalk, and then at the organism levels, it can signal from one tissue to another tissue. Also show you that they can also serve as important communication cues between microbiota and the host. And importantly, at the temporary scales, those metabolites can modulate how long organisms live. So as Yihong mentioned, I recently moved to Janelia and joined the 4D cellular physiology program. So the future goal is to take technology development and mechanism studies to really fully understand those metabolite-mediated regulatory network in time and space and in living organisms. So I already mentioned the people who have been leading those projects. Really want to thank all the current and former members from my lab, our wonderful collaborators who kind of support those technology development and studies in those projects, and the funding agencies who make those research possible. And thank you. So now I'm in the area, so all of you, when you stop by Janelia, let me know. So yeah, we love to see all of you there. Yeah, thank you.